Good evening, all, and welcome to Science Edutech. And today I am going to discuss about the applications of radioisotopes in biosciences, just a basic part. And myself, Vivek Patni, will discuss about the radioisotopes. So, what are isotopes? Isotopes are basically the atoms of a given element with identical number of protons, different number of neutrons, and therefore have same chemical characters but with different number of neutrons and consequently different atomic mass. There are a number of stable isotopes that element possess varies widely. For example, in the case of calcium, there are about 6. In the case of carbon, there are about 3 isotopes. Now, an isotope, how can you write isotope? Okay, so see the diagram here. Okay, now this one isotope is of a sodium. Okay, where superscript to the left of the chemical symbol indicates the mass number, while the subscript to the left indicates atomic number Z. So in this case, a sodium isotope atomic number mass is 23 and the isotope may be described merely as 23 sodium. Okay, whereas 22 is a mass number and 11 indicates atomic number. Now, what is a radioisotopes? What is a radioisotopes? The atoms have same number of protons but different number of neutrons in the nucleus. Because like the charges of the protons repel each other, there are always forces trying to push the atom nucleus apart, and the nucleus is held together by something called binding energy. So this is what a radioisotope is. Now, what is radioactivity? The atoms with unstable nuclei which are constantly changing as a result of imbalance of the energy, within the nucleus is called basically a radioactive. So when the nucleus loses a neutron, it gives off energy. And is said to be a radioactive. And the radioactive energy is released and the matter results from the changes in the nucleus of an atom. So this is just plot showing relationship between atomic number and the number of neutrons So in this case, atomic number Z, okay, and number of neutrons, okay, number of protons, and in this case the solid green line represents neutron to proton ratio. Okay, this line neutron to proton ratio, and red dots represents isotopes, a red dotted line, okay, indicates the isotopes which are stable, whereas the yellow ones, for example this one, are basically the unstable ones. Now, what is half-life? Okay, half-life is basically the 50% loss of the radioactivity. For example, an energy level is reduced to half, okay, and that is called a half-life of any radioactive material. In other words, or it may be also said that the time taken for half the atoms is a quantity of radioactive substance to disintegrate is known as half-life, and it is denoted by T half. Okay, T indicates time and half means half-life. Now, what are the types of radioactive decay? Okay, it is the alpha. Okay, as these radioactive isotopes basically emit different types of radiations also. Okay, alpha, beta, gamma, and X-rays. These are some of the examples of the types of radioactive decay. What are alpha radiation, or what is alpha radiation? They are basically non-penetrating ones. Beta are somewhat penetrating ones. Okay, gamma and X-rays are highly penetrating radiations. Now, the rate of radioactive decay. This graph shown here indicates the rate of radioactive decay. Okay, and the original percentage of percentage here, which is 100 basically. Okay, is the time, as the time goes, okay, it results in the 50% decay. Okay, or T half-life. Okay, that is the time required to decay half of the, or to disintegrate an radioactive substance. Okay, now some of the examples which are used in biological sciences: radioisotopes such as hydrogen, 3 hydrogen, carbon, sulfur, phosphorus, iodine. Okay, these all have different time interval. Okay, that is T half. Okay, for example, hydrogen has about 12.3 years, carbon has about 5,730 years, and 131 iodine has about only 8 days. So different types of radioisotopes have different half-lives. Now the applications. Okay, there are numerous applications of radioisotopes, but in order to use radioactive isotopes, various techniques are used in order to trace the radioactive isotopes used in an experiment. For example, one of them is tracer technique. Okay, another system is a Geiger-Müller counter. Now, some of the examples of the applications of the radioisotopes: medical uses, agriculture uses, in elucidation of metabolic pathways, pharmacological studies, enzyme studies, radioimmunoassays, as well as in the case of forensic science. So basically, these radioisotopes and their formulations have variety of different applications. For example, in the healthcare, they are used in therapeutics, diagnosis, and in the case of let's say agriculture, okay, can be used to check proper distribution of insecticides. Okay, in the case of metabolic pathways, we can elucidate the pathways using the radioactive isotopes. Similarly, there are many number of applications. Okay, thank you everyone. If you want to download the presentation and if you want e-material, okay, please download the Science Edutech app from the Google Play Store for the presentation and a more detailed presentation. And also subscribe my channel, YouTube channel, and also share the Science Edutech app with your friends who are studying any of the life science subject or biological science subjects. Okay, and also share, like my YouTube channel. Okay, thank you very much.